everyone. This week we're going to talk about microbial growth conditions. Specifically, we're going to talk about osmotic pressure. Okay, so we're going to talk about osmotic pressure and we're going to discuss how that osmotic pressure is actually going to affect the bacteria growth. So let's just kind of refresh our memories about the term osmosis. What is the term osmosis? Osmosis means the movement of water. So when the water moves into and out of the cell, that is known as osmosis. Now here's an image of a bacteria cell. Here's the cell wall represented by that brown layer right here. And then of course here's the plasma membrane represented by that yellow membrane on the inside. Now bacteria require water for survival. And so therefore water is going to have to make its way into and out of that bacteria cell. So let's go ahead and take a look at this environment. So let's say here's a bacteria cell and it's placed in this environment. What is this environment called? It's called the isotonic environment. So this term right here, you guys, it's explaining to you the condition outside of the bacteria, not inside the bacteria. Okay, so isotonic, it's again explaining the condition outside the bacteria. Now, what does iso mean? Iso means the same. Okay, so iso means the same. What is the same? The amount of solutes. Okay, so the amount of solutes outside the bacteria is equal to the amount of solutes inside the bacteria. So when the amounts of solutes, those solutes could be, sodium ions, chloride ions, okay? So when the amount of solutes outside of the bacteria is equal to the amounts of solute inside of the bacteria, and so they're both equal, we refer to the environment outside as isotonic to the bacteria. Okay. Now, if the amounts of solute in and out are exactly identical, it's exactly equal, let's say 1% over here, 1% outside, 1% inside, that means that the water concentration is also exactly identical. Okay, so the water concentration outside the cell and inside the cell is also identical. So would the water move? Yes. You might ask, like, why would the water move since the amount of water inside and outside is exactly equal? Water still likes to move, but do you see how the arrow, it's going in and out? So the water is actually going to move into and out of the cell at an equal rate. And so most bacteria out there, because there are exceptions that we're going to talk about, but most bacteria out there, they actually prefer living in an isotonic environment, because when they live in an isotonic environment, nothing is going to distort their shape or like their size, because the water is going to enter and exit the bacteria cell at an equal rate. Okay. Now let's take a look at this condition right here. Here is the bacteria cell. Okay, now the black dots are representing the solutes. Okay, so notice how outside the bacteria there are way less solutes compared to inside the bacteria. See how the bacteria has way more solutes compared to outside? Okay. Now, as I said, we always explain the condition outside the cell. So how is the condition outside the cell? Well, the condition outside the cell, as you can see, it has less solutes. So how can I say it has less? What's the word for less? Hypo. So hypo means low. So hypotonic environment means that this environment right here, it has way less solutes compared to the cell. Okay, so if there is a bacteria, and this bacteria ended up in a hypotonic environment, how is this bacteria, what's going to happen to this bacteria eventually? Okay, so we're going to talk one more time about osmosis. We're going to talk about how is the water going to move. Now, is the water going to go into and out of the cell at an equal rate? No, we said that's going to happen if it's in an isotonic environment. Right now, it's not in an isotonic environment. It's in a hypotonic environment where the amounts of solutes are very low on the outside compared to the bacteria cell. And so do you see how the arrow is representing the water movement, by the way. Okay, so notice how the arrow is rushing into the cell, meaning that lots and lots of water are actually rushing into the cell. Why would the water rush into the cell? Water likes to move from a region with low solutes to a region with high solutes. Okay, you might think, like, again, why would water like to do that? Water likes to dilute things. And so notice how the inside of the bacteria right now has more solutes. So water is going to rush inwards, okay, to try to dilute the inside of the bacteria. Okay, so water, again, you guys, likes to move from the low solute concentration to the high solute concentration. Okay, now eventually what's going to happen to the bacteria is it's going to take in too much water, right? Because all of that water is basically gushing into the bacteria. And eventually the bacteria, as you guys can see, do you see how right here it's basically bursting open? So the bacteria bursts open because, again, the pressure is too much and all of that water is rushing, right? High osmotic pressure, all of that water is rushing into the cell, and so the cell pops, okay? And we refer to that as osmotic lysis, okay? So osmotic lysis, that's basically when a lot of water enters into the bacteria. Again, why is it entering into the bacteria? Well, it's entering into the bacteria because the bacteria is in a hypotonic condition, meaning that, again, outside, as you guys can see, there is less solutes. Okay, and so therefore, as I said, water likes to go from the region with low solutes into the region that has high solutes. See how right here there are lots and lots of solutes, and so eventually what's going to happen is osmotic lysis, where basically the cell is going to pop open. Okay, now what if we have a bacteria cell? Basically the scenario is now flipped. Let's say that this bacteria right here is found in an environment where there are lots and lots of solutes. Do you guys see the dots again representing the solutes? Notice how the amounts of solute outside is way more compared to the amounts of solute inside of the bacteria. Okay, how can I, what word should I use to say that I have lots and lots of solutes or a high amount of solutes outside the bacteria? We use the term hyper. So hyper, right? Hyper means high. So hypertonic environment means that this environment has way more solutes compared to the inside of the cell. And in this case, it's the bacteria cell. Okay, we just said water likes to move from the low solute, okay, concentration to the high solute concentration. So take a look at the arrow, take a look at the water. How is the water going to move? Is the water going to move at an equal rate into and out of the cell? And that would be absolutely no, because if it moves into and out of the cell at an equal rate, that means the bacteria was placed in an isotonic, where the amounts of solute in and out is equal. Okay, now is the water going to rush into the cell? Again, that's another no, because for the water to rush into the cell, that means the amounts of solutes are much lower outside compared to inside of the bacteria cell. So how is the water going to move? The water is actually going to rush out of the bacteria. So the water is going to rush out of the bacteria, okay, at high rates. Why? Because as I said, water likes to dilute things. So outside there is a high amount of solutes, and so the water is going to rush from the low solute concentration to the high solute concentration. So what's going to happen is the cell is going to start losing a lot of water. Okay, and so what ends up happening is the plasma membrane starts to shrivel up. Now the cell wall, because it's pretty tough, it's really not going to shrivel. Okay, it's a tough layer, but the plasma membrane, as you can see, it's going to start shriveling because it's again, now the cell is losing lots and lots of water. And what do we call that? We call that plasmolysis. So plasmolysis, that's when the cell is actually losing. Okay, so let me fix this real quick. So plasmolysis, that's when the cell is losing lots of water because the cell has been placed in a hypertonic condition. As I said, a hypertonic condition means outside, as you guys can see, there are way more solutes, right? See, you can see way more solutes compared to inside of the cell. And so therefore, the water is basically going to rush out and the cell is going to lose water, and that's called the plasmolysis. Now, as I said, most bacteria out there, they prefer to live in an isotonic environment. Why? Because as you can see, with an isotonic environment, the water enters and exits the cell at an equal rate. But there are exceptions to that rule, and some bacteria actually prefer a hypertonic conditions, and so we're going to talk about those exceptions in just a minute. And so right here, we talked about isotonic, hypotonic, and hypertonic environments. By the way, when a cell is placed in a hypotonic or hypertonic environment, and again, this is not its preferred environment, right? We're not talking about those exceptional ones. Let's say that a bacteria which prefers to be in an isotonic environment is now placed in a hypotonic environment. That is actually physical stress, okay? That's actually stressful. It's a stressful condition to be placed in, okay? What's going to happen in a hypotonic is, as I said, the bacteria is going to intake lots of water. Now, gram-positive bacteria actually do much better when they're faced with physical stress compared to gram-negatives. And the reason why, it's because gram-positives have a much thicker peptidoglycan layer, much thicker cell wall layer, so they tend to do much better compared to gram-negatives when they're faced with such physical, stressful conditions. Okay, now before we talk about the exceptional bacteria, the ones that do not prefer isotonic, let me just mention one thing, and that is when we culture bacteria in the lab. Remember how we said when we culture bacteria in the lab, we either culture bacteria in broth, medium, or on slants, on plates, or in a deep. And remember how we said those type of media, the amount or the concentration of agar varies. So broth has 0 agar, and then comes the deep, and then comes the slants and the plates with the highest amount of agar. And we kind of, gave percentages, and we said like the agar in the slants, maximum we add about like 1.5% agar. Now, the reason why we don't add a lot of agar, like, why don't we add way more, agar than, let's say, 1.5%? And that's because we don't want the condition, within the plate to actually be hypertonic. Because when we culture the bacteria, if the amount of solutes, right, because we added a lot of agar, the amount of solutes is now super high, what's going to end up happening is the bacteria is going to go through plasmolysis, and the bacteria is going to lose a lot of water. Okay, and so that's one reason why we don't add a lot of agar, on our plates or slants. Now let's talk about the bacteria that don't like isotonic environments, and they actually like um, high salty or high sugary conditions. So let's first talk about halophiles. Halophiles are actually bacteria that require very high salt concentrations. They love to grow in a condition that has 30 to 35% of salt, and that's a lot of salt. And some of them actually, prefer a bit less, so I mean for 15, okay, so 15 to like 30%. So some of them prefer like a bit lower, 15 to 30%. Some of them prefer like a bit higher, 30 to 35. But I mean, in both cases, that's still so much salt. And the bacteria that love a lot of salt, we refer to them as halophiles. And they are able to talk, like, they're just going to be fine, and they're not going to go through plasmolysis or anything. This is actually the condition that would be best for them. Okay, and that's why, if you think about it, when they want to preserve food, they add salt to it, right? So they kind of like cover it. When they want to preserve like meat, they go ahead and they cover up that piece of meat, or if they want to preserve fish, they go ahead and kind of cover up the whole entire fish with like tons of salt. And that's because not a lot of bacteria can tolerate that high salty concentrations, except of course the halophilic bacteria. They are the ones that prefer high salty concentrations. Now, halotolerant, okay, notice how I said tolerant, not phile. So tolerant, they can tolerate, okay, they can tolerate moderate salt concentrations. We're talking about like up to 10% salt. Okay, they can tolerate up to 10% salt, but they can actually, they usually prefer no salt. Okay, so they actually grow better with no salt whatsoever. But if they are placed in conditions where there is, salt, they can, survive. If it doesn't exceed, as I said, like around 10%. And those we would refer to them as halotolerance. Now there's a term right here, osmophiles, or osmophilic bacteria. By the way, you can either say osmophiles or osmophilic, halophiles or halophilic, okay? So osmophilic bacteria, those are the ones that grow in high sugary concentrations. Now actually, a lot of times though, when you come in, read the definition of osmophilic bacteria, they would tell you, well, those are bacteria that prefer high solute concentration. Whether those solutes is, whether those solutes are salt or whether it's sugar, okay? So if that's the case, osmophiles can tolerate high salt or high sugar, then technically halophiles are also osmophiles, correct? So osmophilic bacteria, those are the ones that would grow in basically high sugar concentrations. For the virtual lab for this week, you are given 3 different types of bacteria, and then you're given 4 plates. Each plate has a percentage of salt. Okay, so this one has like 1% salt, this one has 2.5% salt, this one is 10% salt, and then this one is like 25% salt. And so you're going to go ahead and inoculate the 3 bacteria on each plate. So each plate gets so you can divide the plate into like 3 quadrants and inoculate the bacteria on each quadrant. And then you're going to go ahead and take a look at the plates after, of course, you incubate them. And based on, whether you see growth or no growth, you would be able to tell whether that bacteria is, a halophile, or whether it's a halotolerant, or whether it's a non-halophile, again, like it doesn't, can't do well at all in high concentrations of salt. So that's the whole point of this virtual lab. And of course, this is all back to the whole idea of osmotic pressure, and how well would bacteria do in different conditions of salt, and how the water is going to go ahead, and move into and out of the cells. And basically, when bacteria cannot tolerate at all the conditions that they are put in, well, the bacteria eventually is going to die, okay? And then right here, you're given you're given 4 plates. So let's say this one has 1% salt, 6.5, 10, and 25, right? So we went ahead and we kind of, added a lot of salt, as you can see, just to, just to check, okay, just to check, the bacteria. And you can see that each plate, we've inoculated those 3 types of bacteria. And you can see that, for this plate, for example, you can see that E. coli and S. aureus, you can see the colonies, okay, but do you see how right here for the H. selenarium, there is no growth? So you're going to go ahead and take a look at all 4 plates, and then after you take a look at all 4 plates, you're going to decide, which one is a halotolerant, which one is a halophile, a non-halophile, etc. Okay.